once again, my name is Maria Mendoza, and today we're going to be going over worksheet 9. I'm not allowed to give you the answers, but I did find pictures in the book and information in the book that I'll try to explain so that it's easier for you to understand what is going on. In problem 1, the membrane forms a selective barrier for the passage of molecules. Explain briefly. So A, what kind of molecules can cross the membrane unassisted, and what kind of molecules need a protein carrier to cross the membrane? So this right here is your lipid bilayer. It is hydrophobic, so it makes sense that small, uncharged, and hydrophobic molecules can cross unassisted. Everything else would require transport. So B, why is the permeability of tryptophan at pH 7 significantly smaller than the permeability of the related compound indole? So the first thing I like to do is draw your molecules. Every time they tell you to compare two different molecules, go ahead and draw them so that you can visually see what is happening. And then they're talking about permeability. So what can cross unassisted? Small, uncharged, and hydrophobic. So here you can see tryptophan at pH 7. The carboxylic acid has lost its proton, so there's a negative charge. And at pH 7, the amino group has not, so it has a positive charge. Whereas here at indole, there's no charges. So this should be enough information to help you answer these questions. For 1C, ion channels and transporters allow the selective passage of molecules through the membrane, but they have very different mechanisms. What are the differences between ion channels and transporters in terms of transport rate, saturation, and gating mechanism? Okay, so for this question, figure 11-27 in your book will help you. I also wrote down some information that I found in the paragraphs above and below this figure. So here we have the ion channel has a single gate, and the transporter is a pump, and it has alternating gates. So here for the ion channel, you have one gate. Ions flow down their electrochemical gradients. The limitation is the maximum rate of diffusion. The rates of movement are orders of magnitude greater than through transporters. So this one's a lot faster. And you cannot saturate it. There's only one little gate, as you can see here. And when it opens, then ions can travel down their concentration gradients. And then for the transporter pump, you have two gates. So you can see one gate here and one gate here. And only one gate can open at the same time. So yeah, we have two gates. Only one gate can be open at a time. The substrate can be an ion or a small molecule. The movement is limited by how long it takes one gate to open and close, and only then can the second gate open. Active transporters can move the substrate against its concentration gradient, and you can saturate these. Okay, so question number 2. How does the potassium channel maintain selectivity for potassium versus sodium ions? So here, figure 11-45 will help you to answer this question, but I highly recommend that you read a few paragraphs right before this figure and then a couple of paragraphs after this figure. But again, here are some notes that I took from the book. Everything that's in red are notes that I took. So here in figure A, You can see the inside and the outside. In B, so you see 8 transmembrane helices, 4 identical subunits. You have an inner cone which lines the channels, and then you have outer cones which interact with the lipid bilayer out here. So you have inner cones and outer cones. For B, you have the central channel is only wide enough for 1 potassium ion to pass. So that's the potassium ion right here. So you're looking at it from above. Okay, so then in this picture you have the inside and you have the outside. Here we have potassium with hydrating water molecules. And then you have large water-filled vestibule allows hydration of potassium. So it goes into this channel here. Okay, and then the reason that this works for potassium is that this backbone carbonyl right here in red, all of these right here, they form a cage that fits potassium perfectly, and it replaces the waters of hydration. So that's why these waters can be removed and potassium will enter right here perfectly. And then the carbonyl oxygens form that cage of hydration, replacing the waters of hydration. And then here you have alternating potassium sites. They're blue or green. These ligands can interact with 4 potassium ions, but not sodium ions because sodium is too small. Here you have a helix. It's a dipole. It stabilizes potassium. Here it's positive and negative. Yeah. Okay, for number 3, what is the difference between passive and active transport? So here I have a summary of transport types. Passive. Some types of transporters only speed up the transmembrane movement of solutes down their electrochemical gradients. And some examples are ionophores, ion channels, and passive transporters. Active. Other types of transporters can pump solutes against their concentration gradients using ATP or a gradient of a second solute to provide the energy. Okay? Provide an example of a transport protein involved in the passive transport of potassium and one protein involved in the active transport of potassium across the plasma membrane, and explain the direction of flow of potassium ions in each case, from inside the cell to the outside or from the outside to the inside. Okay, there's a lot of examples in your book, so I'm not going to go there. You just find a figure and that'll be your example. What would happen to the electrochemical gradient if potassium if valinomycin is added to a cell? Okay. Oh, so here figure 11-24 summarizes the various types of transport mechanisms discussed in this section. So learn these. Okay. Explain for each of your examples if the transport is uniport, symport, or antiport. So once you find your examples, then you look at the definitions for each of these 3. Uniport means that you only carry one substrate. So here it is, uniport, one substrate. Symport, you carry 2 substrates, and they're moved simultaneously in the same direction. So substrate 1 and substrate 2, and they're both going in the same direction. And then you have antiport. You have 2 substrates that move in opposite directions. Okay? So what would happen to the electrochemical gradient of potassium if valinomycin is added to a cell? So again, figure 11-42 will help you answer this question, but I highly recommend that you read a few paragraphs before and after this figure, because the book does a really good job of explaining exactly how to answer the question on your worksheet. Valinomycin, a peptide ionophore that binds potassium. In this image, the surface contours are shown as a yellow envelope, through which a stick structure of the peptide and potassium ion in green are visible. Let's see, the oxygen atoms that bind potassium are part of a central hydrophilic cavity. So these are the oxygen atoms. And so you know they're a little bit negative, and then the potassium is positive. So they want to interact with each other. And this is your hydrophilic cavity. Hydrophobic amino acid side chains in yellow. So these are the hydrophobic amino acid side chains. They coat the outside of the molecule because the exterior of the potassium valinomycin complex is hydrophobic. The complex readily diffuses through membranes. So because valinomycin is hydrophobic on the outside, it's able to just flow through the membranes, through the lipid bilayer, and it carries potassium down its concentration gradient. The resulting dissipation of the transmembrane ion gradient kills microbial cells, making valinomycin a potent antibiotic. So this is really important, especially to answer your question. But like I said, I recommend that you read a few paragraphs. Okay, number 4. When an uncharged molecule moves from a concentration of 10 to the negative 4 molarity to 10 to the negative 2 molarity, is the process spontaneous? Is it at equilibrium? Or does it require an input of energy? So whenever I get numbers like these, I like to rewrite them. So you basically have 10 and you move 4 decimal places to the left, 1, 2, 3, 4, and you put your dot there. So this becomes 0.0010 and 0.10. So this number is smaller than this number. So you're saying that, well, actually, let's go here. When something moves from a higher concentration to a lower concentration, it is said to move down its concentration gradient. Here you're moving from lower concentration to a higher concentration. So based on what you've learned about passive and active transport, you should be able to answer this question. Okay, number 5. E. coli cells transport xylose across the cell membrane via a symport system that uses the free energy of a proton gradient. The intracellular pH is higher than the extracellular pH. A. Xylose moving up or down its concentration gradient. Okay, so what is happening? Is xylose moving up or down its concentration gradient? Is it moving into or out of the cell? So here they're giving you a clue when they tell you that the pH inside of the cell is higher than the pH outside of the cell. Remember that low pH means more hydrogen ions, and high pH means less hydrogen ions. You should know how a symport system works. Also notice that there is more xylose inside the cell than outside. Still, xylose is being brought into the cell. This should be enough information to help you answer the question correctly. complete the xylose transport system. So here you just have to write arrows. Where is hydrogen moving? Is it in this direction? Is it in that direction? And then in what direction is xylose moving? Is it in this direction or is it in this direction? Okay, question number 6. Energetics of the sodium-potassium ATPase. A. Assuming a membrane potential of minus 70 millivolts, so the inside is negative, a concentration of sodium inside the cell of 20 millimolars and 145 millimolars outside of the cell. In which direction will sodium move spontaneously? So here it's just spontaneous. So that means down its concentration gradient. So it'll just go from high to low concentration. And that should be enough information for you to be able to answer that question. Now, let's see. Under the same conditions, calculate the total free energy change for transporting 1 mole of sodium out of the cell at 37 degrees Celsius. What is the change in free energy due to each, the chemical gradient and the electrical potential? So you're going to be using this equation here. Okay, so this value here is the chemical, and this is the electrical. Okay, that should be enough to help you answer that question. Now, here is some information that I found in the book for you. Where is this pump found? This sodium-potassium ATPase pump. It's found in the plasma membrane. It is a co-transporter. It couples the phosphorylation and dephosphorylation of a critical aspartate residue to the simultaneous movement of both sodium and potassium against their electrochemical gradients. The overall goal of this pump is to maintain the inside concentration of sodium low and potassium high. Every time the cell converts 1 molecule of ATP into ADP and phosphate, the transporter will move 2 potassium ions in and 3 sodium ions out. This type of co-transporter is electrogenic. It creates a net separation of charge across the membrane. In animals, the membrane potential will be minus 50 to minus 70 millivolts. 25% of the total energy consumption of a human at rest is invested in this reaction. Z is equal to charge. So sodium has a plus sign here, so that's just 1. If it was a plus 2, then this number would be 2. And now I'm going to read you the figure 11-36. Okay. So role of sodium-potassium ATPase in animal cells. This active transport system is primarily responsible for setting and maintaining the intracellular concentrations of sodium and potassium. And like we said, we want to keep sodium low and potassium high inside of the cell, right here. And you can see that sodium is 12 millimolar and potassium is 140 millimolar. Okay. The electrical potential across the plasma membrane is central in electrical signaling in neurons, and the gradient of sodium is used to drive the uphill co-transport of solutes in many cell types. Okay. So learn to use the formula with this example from the book. Yeah, if you didn't know how to use this formula here, this example from the book is really good to help you figure out how to use the formula. Let's see here. Many pumps are members of the P-type ATPases. If you discovered a new enzyme with similar function, what reaction intermediate would help convince you that your enzyme was a member of this family? So you have to know what is a P-type ATPase pump. So this is a great figure in your book. Um, and over here is the legend. So they didn't fit on the same page. So go ahead and read this. Figure 11-34. The general structure of the P-type ATPases. P-type ATPases have three cytoplasmic domains, A, N, and P, and two transmembrane domains, T and S, consisting of multiple helices. The N nucleotide binding domain binds ATP and magnesium, and it has protein kinase activity that phosphorylates a specific aspartate residue found in the P phosphorylation domain of all P-type ATPases. The A actuator domain has protein phosphatase activity and removes the phosphoryl group from the aspartate residue with each catalytic cycle of the pump. A transport T domain with 6 transmembrane helices includes the ion transporting structure, and 4 more transmembrane helices make up the support S domain, which provides physical support to the transport domain and may have other specialized functions in certain P-type ATPases. The binding sites for the ions to be transported are near the middle of the membrane, 40 to 50 angstroms from the phosphorylated aspartate residue. Thus, aspartate phosphorylation and dephosphorylation does not directly affect ion binding. The A domain communicates movements of the N and P domains to the ion binding sites. In Figure B, you will see a ribbon representation of the calcium ATPase circa pump. ATP binds to the N domain, and the calcium ions to be transported bind to the T domain. Other P-type ATPases have domain structures and presumably mechanisms like those of the circa pump, shown here. Shown here are sodium potassium ATPase, the plasma membrane hydrogen ion ATPase, and the gastric hydrogen ion potassium ion ATPase. Okay, so that should be enough information. Honestly, here I also recommend that you read a couple of paragraphs before and after. The book does a really good job of explaining everything.